So this is week 13, and the next week, week 14, is your presentation time. So that will be the 23rd April. We have 8 students. So Sarah, you are here in this group next Tuesday here, okay? Sarah Wolf will be Thursday next in that room. So you just on your mind to prepare which room you will stay, okay? Carissa will be the last one. So that's the setup. I think for 8 students, we should have enough time for 8 minutes talk, 2 minutes questions. And those are the criteria, and the description of those are based on your presentation is about a proposal, a study you haven't been doing, but you propose what you want to do. But if you want to present some preliminary results, you just change some terminology, but in general, you have significance, innovation, approach, and also your personal performance on the podium here. Okay, so we just spend some time to describe this. So this week, we will focus on graph theory and the network analysis. Again, this is a very large topic, and some of those lectures can be expanded to a whole semester theory and practice consideration, and there are a lot of topics that we should be able to cover. But that's an outline for today. So we are going to look at the definition, network, and the mathematical description of graph, and then we will go through a theory of this called centrality measurements for the nodes, of importance of the nodes in the network. And then we will mention WGCNA algorithm. The reason is this very popular network analysis package has been widely used. If you look at the citation of this paper, describe this method, it maybe have, I don't know, estimation 5,000 citations or more. And then we will mention the community of modulity detection algorithm, and then we move to single cell gene regulatory networks. And also Genie3 is a package using random forest algorithm to predict the association between genes to make networks. And then that's in the category of this CART algorithm. We will mention multi-layer networks, okay? And then at the end, I will mention the single-cell tenfold. The toolbox, there are a series of the methods we developed and centered around networks. So this is a typical procedure that you can use. This example is use functional MRI time series data to construct a network. So you sample signals from different regions of brain, okay? So that's indicate orange and this blue, 2 signals cross time. And then you look at these 2 points and those 2 signals correlation, then you make a correlation matrix. This matrix contains many sampling points, but there's only 2 has been indicated in this small example. But each point here is a pair of signals. You can have a lot. Then you transform this correlation matrix into an adjacency matrix. Adjacency matrix is the network itself. Once you have this network, you need to know what to do next. This is called functional network analysis. You should, after this lecture, you should have many methods you want to apply, okay? So this is very clear example, but when we think about the gene regulatory network without single cell data, you have to think have you ever used network analysis in your not single cells, such as the bulk RNA-seq or microarray example data sets before? So how did you prepare that if you don't have this time series data? Anyone have any experience to make co-expression network from expression data? Yeah, we probably will mention that how you traditionally, conventionally, without single cell data, how you make network. But you see those papers, right? Anyone have read those papers? Gene expression analysis using bulk RNA-seq and build a co-expression analysis network. We will touch base on those later. So basically, okay, graph network are the same thing, and they just use this terminology in different fields of the in mathematical graph theory. They use graph in computer science. Maybe people use networks. When you talk about the graph, you have vertices and the edges. And then same thing in network, they call the nodes and the links. But sometimes those terminology can be used interchangeably. So here is an example of a small network contains 5 nodes. And they're linked in this particular way, so form this spatial topology. You can express this structure using a matrix A. So this is called adjacency matrix because you have 5 nodes. So this A is a 5 by 5 squared matrix. Adjacency matrix should be squared matrix all the time. Then if you have, so each row or each column is the index of your node. So the 4th row will be the node number 1. And its connection with other nodes will be indicated by these non-zero values. So for node 1, it's linked with node 2. So that's why this position 1, 2 have a 1 here. Node 1 not linked with any other nodes except 2. So you have all zeros. Okay, it's not also self-linked. So there's no 1, 1 position is 0. So same thing, you just follow the next one, and the 2 is linked with 1, 3, and 5. So you have 2, 1, 2, 3, 2, 5 position of adjacency matrix are non-zero. Okay? So once you have this description, by the way, this is a undirected, okay, and also unweighted, because 1, 2, 2, 1 are the same for this. There's no direction. So that's why you have a symmetric squared matrix, adjacency matrix as a representation of the topology. And also, all the links are same weight, so you just put a 1 indicate they're linked. If it's weighted network, you can see, okay, the link between 1 and 2 is strong, and then you put 2 rather than 1. Okay, you double the weight, for instance. Okay, so just imagine once you have a network like this, okay, this network, I don't know how many nodes, 5, 6, 7, 8, 9, 10, 11 nodes, this small network. So what kind of question you can ask? The centrality measures are a series of designed scores to indicate the importance of each node. Okay, so those centrality value can be assigned to every single node in the network. Depends on your definition of what type of the centrality you use. You describe the importance of the nodes in a different way. So most commonly used centrality is called a degree, just how many directed partner linked with you. So in this purple node here, okay, the purple node linked with 1, 2, 3, 4 different immediate neighbors. So you have a degree equals 4. And this green dots have a degree 3, right? And this one have 1, 2, 3, 4, 5, 6. The right one have a degree 6. So this is the simplest way to describe the importance of a node. It's intuitively easy because you have a network node and linked with many partners, then looks like you are important in the center. This is not the only way, and I can show you immediately. And there's another way called closeness centrality, okay, to describe this. Let's see what's the definition of closeness centrality. So this is described as looks complicated, but all the formulas in the network theory is relatively easy because every parameter here, they're physical or this direct intuitive meaning in the network. For instance, what this D is? So D is the distance between node X and Y. All right? So now we are talking about to compute the closeness centrality for X. So for example, we are saying that X is A, so in this network. So you replace X with A, and then Y is all the other nodes other than A. So you measure the distance from A to all the other nodes. So from A to B, there's one step, distance 1. And then from A to C, 2 steps, distance 2. A to D, 3 steps. A to E, 4 steps. So you have A to all the other nodes, you have the distance or length of the path from 1, 2, 3, 4. Okay? You sum all this distance together and 1 over it. So this value is so-called closeness centrality for A. Easy, right? Then you move to another node, say B. Now you replace X with B. Okay, you look at the B's distance to A, to C, to D. And in that case, you compute the total distance from the B to all the others and all 1 over it. And you get a value 0.14. So you repeat, do the same thing for C, D, E. Okay? Yeah. B to A will still be 1. Yeah, although the same. So at the end, you look at their values. What you found is C have a larger value. The reason is C is in the middle of this network. It's close to, relatively close to most others. So that's why 1 over the total will become larger because the distance is small. And also the symmetric A and E has the same topology position, so you have same closeness centrality values. And the B and the D are the same. All right? So this introduces another way to describe the importance. If you use a degree, C, B, D are the same. They all have 2 partners, but they're not considering the global distance from C to all others, but this closeness centrality tells you C is in the middle of the network, is more centered than B and D, okay? For this simple network, it's easy to see, okay, C may be important because it's linked to all others in the middle, but you have thousands of nodes in a large network that you need to have those computed matrix to tell you which one is the most important. That's one, the second. And also important, you can define in different ways, okay? Define the central. So this is a different ways to call harmonic centrality. If you look at the formula, it's almost the same, just slightly arrangement differently. So rather than 1 over the summation of all the distance, here is 1 over first distance plus 1 over second distance. So at the end, it becomes the harmonic mean of the all distance. So this is called harmonic centrality. So if you look at the computation, so to compute harmonic centrality for this node, you look at its distance to all other nodes in the network. It has to be connected. Here is a separated component we will not consider because the distance from this to here is infinity. So we don't consider, only consider the distance from one node to this all fully connected so-called component. Then you put all this distance in the 1 over them, then summation, make summation together, and you got 2.5. So then you repeat for next node, do the same thing. So this is just 2 examples for you to have some idea of what we're doing here. So next, I'm going to introduce an index called the Kazu index. It's also a centrality measurement. It's slightly more complicated than that, okay? So that's why we need to have this illustration here to know what's the power of adjacency matrix stands for. On your left is directed network, okay? On your right is a matrix called M. And this network has 6 nodes. So this M adjacency matrix is 6 by 6. And M is not symmetric because the network has a direction. So from 1 to 2, there's no connection. You cannot go from 1 to 2, but you can move from node number 2 to 1. So that's why you have 2, 1 position is 1, but 1, 2 position is 0, okay? That's not hard. If I cover this, you should be able to rebuild from that to here, okay? Either way. So now I'm going to let you to compute M times M. So this is a multiplication between 2 matrices. Can you do that? You should be able to because this is a squared matrix. N by N is equal to 6. So you can apply this matrix multiplication to do this matrix multiplication. You have M here. Remember? So N by N. The way I showed you, you should put another matrix here. It's also N by N. So its product should be in this corner here. Right? So this should be M power 2. So the dimension of this is still N by N. So that's N by M times M. Okay, so this is the results of the multiplication from that to power. So what this tells you is by So here, for example, there was 0 from 1 to 2. There's no way you can go back, okay? But after the multiplication itself, it becomes 2. There is a meaning of this. This tells you there are 2 different ways from a 1 move to 2 by 2 steps, okay? So this is also a count, and this indicates how many steps. And this position tells you from which node to which node. So when you have a 2, that indicate they're linked by 2 steps. So the 2 indicate you can move from 1 to 2 by through 3. So 1, 3, 2. So there's 2 steps, the one way. Another way should be 1 to 4 to 2. Okay? So this number tells you with 2 steps how many different ways you go from one node to another. So this 3 here, okay? If you count, this will be 1, 2, 3, 4. From 1 to 4. Yeah, so 3 means from 1 to 4 by 2 steps, okay? So we can try from 1 to 4 by 2 steps. You can go, should be 4. So you can go from 1 to 3 to 4, so it's one way, okay? You can go from 1 to so this should be from 4 to 1 maybe. Yeah, so I believe this number here should be 4 from node 4 to 1. 4 to 1. By 2 steps, you have 3 different ways, okay? The first way will be 4, 6, 1, 4, 5, 1, 4, 2, 1. All right? So this matrix tells you indirect ways by fixed number of steps from one node to another, how many different ways you can have. So this gave you an impression of the connectivity between 2 nodes. Although they may not directly linked, but you can find alternative way and a little bit longer, okay? So the power indicate how long you will consider the steps, and the number in the product will tell you how many different ways you can go. So causal index will consider the first power, okay? So this will be adjacency matrix itself, the direct link from one node to another. It will also consider 2 steps connectivity between 2 nodes, and also 3 steps until a large number. So this causal index consider direct link, 2-step links, 3rd-step links. So this overall potential from one node to another. And it will also consider how many different ways you can go. So you distribute your connectivity power from one node to another equally. If you have 3 different ways to go, you can weight each pathway in 1/3, because you have 3 different ways to go by 2 steps, or 4 different ways by 3 steps. So this is a huge description of the global connectivity between nodes. So this index will consider for each node its potential to link with all the other nodes, and also consider multiple steps and multiple ways. Okay, so now if you consider computation, to compute the causal index will spend much longer compared to the closeness centrality, the simple ones, right? Okay, so this is Any questions here? All right. By the way, this is very important, interesting properties to take power to consider this multi-steps connections between nodes. So if you have the summation of many, so if you plus this with this, and more, you have global description of those influence or connectivity between one node to another. So if you consider this as observed connections in your data, you probably will take exponential of the observed network in order to reconstruct the real connectivity between, because what you observe from one node to another, it could be due to this multi-parse, different ways connections. If you really want to learn the intrinsic connections between nodes, you probably want to derive this using the exponential function of the matrix. Okay, so this is an example to show using different centrality applied to these nodes, you get different kinds of measurements of those. So using high degree, you consider the same network, and you consider those nodes looks more important. The value is high. But if you apply, see, this one, the Kazu index, okay, Kazu centrality, then the D tells you those are the more important because it's consider multi-ways of the connections between cells, between nodes. So at the end, you probably want to create a table for you if you have a network here, okay? You compute all different kinds of centrality. You can try degree, closeness. There are also betweenness, okay? Katsu. So you can compute all this matrix. Then at the end, you can have a table. So this is my node 1. Node 1, 2, to n. So you can have all the nodes, then you have degree values. You can have closeness, you can have betweenness, Katsu. You can have many, many different values for all the nodes. Give you a better definition of the importance of the nodes and using different perspective of the understanding of the importance. And also, this is a very active field. And if you look at this new publication, this may be recent published, and they start to define another centrality, and it's called leverage. And the leverage centrality, if you compute, you follow the formula, you will get higher centrality value for those nodes, those ones. Okay? Rather than those in the middle. So this particular design centrality will find those bridge nodes linked with one huge module with another. And if you break that particular node, for example, you delete this, and then this becomes separated to large modules. So this particular designed to emphasize importance as a bridge between modulities. Any questions? We are moving to another topic. So you know how to do this, define once you have the network. So as I said, this WGCNA stands for Weighted Gene Co-Expression Network Analysis. You can give a search like a Google Scholar and how many citations, how many people has used this to build co-expression network. Again, we need to set back and see, okay, if you have microarray-based or bulk RNA-seq you have those experimental results. How can you make a co-expression network? So what type of data you need to have? So bulk RNA-seq, you have something like this. You have a lot of genes, okay? Gene 1, gene 2. But you don't have a lot of not a cell, so you have several samples. So sample 1, sample 2, sample 3, sample 4, see, okay. This number shouldn't be hundreds, okay? Maximum, you may have 7 or 8, 7. So let's see, as 5. So you have 5 experiment conditions. You want to build a network. The people doing this all the time, but believe me, they're useless because you only have small numbers of samples. If you are doing this comparative design, then you have, for example, you have 1, 2, 3 are one condition. Maybe just add one more sample, so sample 6. And then you have 4, 5, 6, another condition. So you want to compare the higher low. So you have a gene in this condition, gene 1, but in condition here, you have increased value. So you upregulated this gene. In that case, for the profile of gene 1, so for gene 1 expression, you have 1, 2, 3, sample 6, 4, 5, 6, and then the expression value was here, and then upregulated. That's all the information you have, which contrasts between these two conditions. So you say, okay, this is upregulation for gene 1. And then if you found gene 2 have a similar profile, also upregulated, then gene 2 will be, let's see, okay, was here and upregulated. Goes to the same direction. So that's all. You think, okay, they are correlated. Then you believe this gene 1, gene 2 in the network should be connected. This is a terrible design. You shouldn't do that because you only have 6 points, and there's actually 2 conditions. There are many noise. You can get this co-expression up, down at the same time. Okay, So I believe if you apply this to most of the traditional bulk RNA-seq or microarray design, the problem just not well defined. Okay, especially you have hundreds, thousands of genes, and you only have 6 conditions. There are many false positives you can create. You just random pick 2, you get positive and negative correlation all the time. But anyway, so this you do experiment, you don't want to waste your time and resource. Your PI asks you to build a network because other people are doing this. So you just plug that in to give the results. But most of the results have been wasted. That's what my opinion is. So here is a description of what I'm describing. So gene 1, 2 are co-expressed. That's why you put they are positive correlated. Some gene downregulated was highly expressed, maybe lower here. So you have those genes, 3 these green genes co-expressed, co-downregulated. And then you make those connections, say, okay, there are positive correlation between gene 1 to 3, okay, the right one. And there's no correlation between those blue ones, and then negative correlation between 3 green genes. So you start to make a link between those genes and try to identify those co-upregulated, co-downregulated genes at the same time. So that was the idea.
So what is a good input data like set?
For this? I believe there are some conditions and you can somehow get a better resolution. So this design is 2 conditions and with some biological replicates. But if you have really good design, something like this, instead of 1, 2, 3, 4, 5 from same condition, so this could be from a temperature 20 degrees C. This will be sample 25 degrees C. This will be 30 degrees C, 35. So you have some of those conditions which make some covariance, okay, start to make sense. In that case, this 1, 2, 3, 4, those values corresponding to 20, 25, 30, those values, if you have a gene, happen to be time sensitive. So I believe this will be a better situation compared to this.
So in that case, you would just be doing one condition, and you could make another one for a different condition, and then see what happens over that temperature.
Yeah. So I believe those, and this could be the better resolution. So the traditional bulk RNA-seq is you don't have enough samples. Okay, the correlation you see, you compute is not very reliable. I believe somehow this WGCNA become popular because it's applied some of the additional 2 steps in order to help transform this raw correlation matrix to somehow more robust. So maybe some signal has been recovered. So we just say, okay, this is not reliable. If you compute, look, some genes co-expressed, anti-correlated. But what they did is after the construction, this network, they first to transform those values, okay, either by taking absolute value or apply this. So this will be changing this correlation coefficient between -1 to 1 and change to 0 to 1, but it's just rescaling. So to keep there's no negative values. Then they set up a cutoff. So either you have to be 0.75 above, then I will keep that. Otherwise, I will trim off the link between these two genes. So they put some threshold to filter this, or some soft cutoff. So those are very stable, very standard. And then what the special about this method is they borrowed an idea, so-called topological overlap matrix, to transform this correlation matrix into this topological overlap matrix. Okay. So this smooths or smooth out those superior signals between pairwise genes, and try to look at these two genes has to be connected if they have a lot of other supporting information from the neighbor genes, which show the same kind of overlap in their topology. Okay, so let's see. They apply this to smooth out. So this is original correlation matrix, and then if you smooth it out using this topology by 2 steps, then you get something like this. Okay, so they trust this transformed matrix and apply downstream analysis based on that, not directly on the correlation matrix. So what it is? So this is an idea they borrowed from another paper published in Science, okay, 2002, in the Babasi group. What they introduced the topological overlap concept. It's pretty simple, actually. Okay, so you look at the number of the neighbors shared by 2. So once you have a network, they look at the number of shared. Let's use this example. I'm using these 8 small examples to illustrate the equation, and you see, okay, everything seems direct and simple. So if I have this node, so 1, 2, and this 2 has no direct link, so the network looks like this, okay? So this 2 nodes has no direct link, but we are going to compute so-called topological overlap between them. So we look at their direct neighbors. So this node have this direct neighbor, those ones, okay? And the 2 have those ones. They share the 2 common nodes here. So from the equation here, okay, so the equation I have I plus 1, j plus 2. And this n is the neighbor of I. So neighbor of I is neighbor of 1 are those. So neighbor of my first node joined with neighbor of my second node. This is just count. How many? The 2. Okay, so this number is 2. They have 2 shared neighbors. And then I look at how many neighbors for 1. So neighbor of 1 and the minimum neighbor of 2. So minimum. Neighbor of 1 is 1, 2, 3, 4, same as 1, 2, 3, 4. So this is 4, this is 4. So minimum between them is also 4. So 4 is here. And they put the equation say ij, so this is some parameters, okay, to adjust the values. Let's put 1 there, 1 there. So at the end, you have +1 here, +1, you have +1, or 0 maybe. So anyway, at the end, you get 2 minus this. Let's put this 0 here, 2/5. So this is the overlap, the topology overlap between two nodes, giving this topology. So that's the value we compute between 1 and 2, 0.4. Okay. So this is consider only one-step neighbors. You can consider for each node, you consider two-step node neighbors and see whether one to have a shared two-step neighbors as well. So change the formula, the weight of count, slightly different. But same idea, okay? If you consider 2 steps, then the overlap between this becomes 0.67. So instead of using correlation matrix, you compute based on the correlation matrix to compute this topology values, overlap values between nodes, and replace that correlation matrix values with this topology computation values, computed values. Okay, you transform correlation matrix into this topological overlap matrix. So here is topological overlap matrix 1 indicated you consider one-step neighbors. You can move to higher steps, okay? So this is consider 2 steps neighbors, and this considers 3-step neighbors. So you transform your matrix, original network matrix, into a more smooth version, consider not only direct link, but also the overlap between topology of the similar topology neighbors, and smooth out the whole matrix. And then you start to build clusterings from this matrix, okay? You smooth the signal and start to build clusters. And those genes co-express will be clustered together. So that's a classification. If you apply classification algorithm to adjacent matrix directly, you get a lot of noise signal. But here, you apply to this topological overlap matrix, the transformed matrix, then you get very clear, big chunk of those gene sets, co-express networks. That's all. This is the whole idea based on the Baba-Bassi, this idea, okay? But the R package has been so popular, and you see this thousands, hundreds of citations of people using this approach. Okay, to find the co-expression networks, and they start to do a lot of downstream analysis based on this. For example, you can see, okay, which particular subnetworks gene expression correlated best with the different conditions. Let's see the time. Any questions so far? Yeah, I will use my break time to look at the citation of the WGCNA, how many papers I've been using this. So next topic we will discuss about the detection of subnetwork. So within the huge network, do you have subnetworks, or you call them modules? Modules, those something like this. Okay, you have a hub genes, and you have a lot of heavily connected other genes groups, and form these modules. And between different modules, there are links, but the connections between modules will be relatively small compared to the links between nodes within the network. within the modules. So we want, in the social network study, people want to identify those clusters. So what group of people or movies or what kind of books are in the same group categories from those modules. So in the gene network analysis, we want to work out those modules to understand whether some modules associated with particular function. Okay. So the algorithm people mostly use is called the Leuven modulity. So this is based on statistic called Q. You compute this. So this Q based on a formula like this, and then at the end you have a delta function. So this function tells you if you have two nodes, I and J, if they belong to the same community. So the indicator of the C is the community the node J belongs to. Okay. For instance, if I and J are two nodes, they all from the community number 5. So C is 5, 5. Then if this is a 5, this is a 5, okay, this delta function gives 1. So if this value is the same, the function gives 1. If the value is different, this function gives 0. So depends on whether these two nodes are inside the same communities or not, you compute the Q. If they are not in the same community, the Q is 0. All right? So within this same community, you start to compute the weight, the link between these two nodes, how strong they linked, because that's the weight of the link. If this is unweighted, Aij will be 1. And then you minor this normalized, the product of the K. So K is the nodes with all the other neighbors, I believe. So anyway, so this Q will be a modulity score, basically, for this condition, I and J. So you loop through all the Ij pair, okay, to give you the overall modulity level of your network. So this will be hugely impacted by your assignment of the neighbor members. Okay, if you assigned correctly, so the neighbors, they are in the real community, so you see membership ID are the good reflection of the reality, then you get larger Q values. Otherwise, if you randomly assign, they are not real nodes in the same community, then this value will be relatively small. The only you maximize the Q, you get the best solution for your community assignment. Okay, so there are some of the applications to look at the modulity, and so to compare those. So I just threw out some of the papers, all the papers, and if you want to go deeper, then you can take a look. Okay, we should take a break and come back from after 5 minutes, and we start to introduce the single-cell gene regulatory networks. Okay. Okay, so the single cell data actually bring this expression matrix from hundreds of thousands of cells. So that has solved the problem of the small sample size problem. So, and that's the most important contribution, I believe. And also, you can do this for particular given cell type. So you are doing cell type specific gene regulatory network construction if you start from single cell. So that's one of the illustration because we get this matrix from 10x genomics, for example. This is an example data set, the real data looks like, and you have, you can compare, for example, get two genes expression profile out, and they are sampled from many cells. So you can build a beautiful correlation matrix, correlation relationship between two genes. And those values have been using magic to impute. So that's why you see this beautiful correlation between two genes. And then there's nonlinear relationship you can recover from this. You see, okay, these two genes, they're not monotonically increased expression when they are in different cells. And then if you look, if you put in the third gene's expression, so the third gene here is called PRNM1. This gene's expression is indicated by the color, okay, rather than the XY axis values. So this tells us for a group of cells which this gene expression level is relatively high, which are those cells. If you look at the color, it's more yellowish. Well, the other group of cells are relatively lowly expressed in terms of this PRDM1. So if you conditioned on this gene's expression, you see, okay, these two genes, IRF4 and AICDA, they are either positive correlated when this gene expression is high, or they are negatively correlated when this third gene expression level is low. So that's why, because the number of points here, there are hundreds, thousands of cells, so you start to look at those correlations between gene pairs, or even higher high-level relationship between three genes, even higher. So you can start to build a better network compared to these conditions. You have only 6 points for each gene. And those three genes are not random genes. There's genes in this well-studied NF-kappa B regulatory network signaling pathway. And cells are from a B cell derived lymphoblastoid cell line. So everything starts to make sense. You can reconstruct pairwise relationship between genes by looking at multiple genes relationship. And that's real. And we look at all the genes in this given well-studied gene regulatory network, the small network, and it composed of 1, 2, 3, 4, 5, 6, 7, 8 genes. Okay, so all these links, solid links, are the immunology study from animal, from cell lines. At the end, you come up with this model. Okay, this is from a literature review. So every link between two genes may have multiple evidence from animal study, from experiments, from the cell lines, perturbation conditions, so people know those are the close to reality. All this relationship can be recovered from the single cell data, okay? Just one single cell data. We just look at the pairwise relationship between genes, okay? So the first gene with the second gene, they are positively correlated. And then if you look at their the link between them, this from AICDA to batch 2. Yeah, some of this is not a direct link, but you know the correlation. At the end, you can almost reconstruct the whole network. The efforts are different. On your right side, this is one cell line, single cell RNA-seq. On your left side are the decades of the efforts from experiment biologists. They put every resource together, build up this network. But you just one experiment, the power of the single cell can allow you to recover almost the same pathways, or almost the same network. You can also, as I said, everything is cell type specific. Okay, you can have a cell type identification and apply network analysis for any given cell type you are interested in, rather than put them together, build a network which the signals are average across different cell types. But here you know, okay, this link between this gene with gene 4 that only happened in orange cells, not in the tail cells. So they are specific cell type effects. There are many, many advantages, okay? Large sample size. Yeah, so the variability internally have there. Remember, I had this, the variability, the design at different temperature. You have 25, 20, 25, 30, 35. The experiment design in that case try to have the variability across conditions. Without the variability, you don't see the change, you don't have the correlation. But single cell data internally, they have variability already. Cells in different stage, they are moving back and forth. So that variability help you to look at, to check the correlation between genes in a very natural way. We can also possibly collect data at different time points. Okay, even for the single cell, you can have a day 1, day 2, day 3. You can build a pseudo time, so you can have real time theory or pseudo time theory data to help you to understand the dynamic of the regulation. You can also, so here is a review paper talking about apply clustering algorithm. So you have A, B, C, different cell type to do the cell type specific network. You can contrast or compare between network constructed from different cell type or different conditions. You can have a trajectory analysis to build a pseudo time, so you have a time series data allow you to construct multiple networks to follow the dynamics of the regulation. And this paper also gave a list of the method has been specifically designed for single cell data to construct gene regulatory networks. So this is an illustration of the idea to do this infodynamic networks, okay? You know you can do pseudo time, or if you have time points, you can build some real time points data, and then the method that you can use dynamic network inference approach. Otherwise, you don't have those information. Then you can do a static network without considering the change of time. So those are some top popular methods people have been mentioned or used. Again, we are not using any of them. We use our own so-called PC regression built into the single-cell 24-net approach I will mention later. But here, just some other people developed the methods. And there are benchmarking algorithms to compare the results of those gene regulatory network construction algorithms and put them side by side, apply to the same input data, see which one gave you the best results. The problem is this evaluation network gave as a test are small networks that only contain 20 or 30 genes. But in reality, we have the situation in a single-cell RNA. We try to build the transcriptome level of the gene regulatory networks. So those evaluation study has some values, but have a very limited contribution to the field in my term. And they test different methods, say, okay, which one gave you the ROC curve, the best area. So those are accuracy and sensitivity. Some of this overlap, whether the constructed network is similar to what they expect to see. And also, again, in biology, we don't have a ground truth. So I don't know what they believe, which the solution is the best, what's their criteria. But it's better than nothing, but someone has already done this analysis. And this network they used as a ground truth, and this is one. There are 4 different. As I said, the largest network they test contains this number of the genes. Okay. So some other test network even only have this 5 genes. So it's not very useful to as a standard to see which algorithm is better than others. Again, this is summary of results for the data set. So different data sets, different algorithms. Okay. So Gene3 is a network construction algorithm based on the trees. Okay, tree-based method. It's not designed for single-cell data specifically. It has been applied to some of data like this. Okay. So the algorithm itself is a random forest method, a tree-based method. So this random forest, as you remembered, is one of the classification algorithm. You put a binary tree and based on the features and classify data points into group 1, group 2, group 3, something like that. You can also apply this framework to do the regression. Okay, regression means you predict the value of this given input. So you can do this using classification algorithm to do regression because you can consider this classified groups. You can put a lot of small groups, and when you have enough, then the problem becomes a regression. All right. So the whole framework is based on this called decision tree algorithm, classification or regression trees. Okay, the same framework. So what is decision tree is like a binary tree to ask you a question. Okay, your input is the height of this person. And then the first, they want to classify whether this is a man or women. Then you have the height and the weight to describe two parameters to describe one sample. And then the first question is whether you're greater than 180 or less. So yes or no, and then your first classification. Then you have weight greater than 80. Then that's the very simple binary trace to do the decision, to do the classification. And you can consider a more complex or more generalized approach. You have two parameters, x, y, and you have multiple decisions in there, and then you can classify the input sample, the training data, into these different categories indicated by y. So in this 3D is x1, x2, and y is the z-axis. If you have enough this internal cutoffs, then you can refine this landscape very thoroughly. So every single sample you can put in certain given y, depends on the training data. So you can make this landscape really detailed. At that time, your model becomes this trained landscape becomes a predictor or regression tools. Once you have new data comes in, depends on where you move, you're standing either on this plateau or here, or that give you the prediction or regress the values. So this is an example. If you have the horsepower and this wheel base, some two parameters, and those values, the price, are given by this color. And then you can make multiple decisions inside. Then you bin your training data into different small chunks of those areas. And then once you have a new data points comes in, you can predict its price. Okay, so the problem becomes the regression. So Genius 3 is use this framework. It start by looking at this expression matrix, and then start to build the prediction model for each gene. Okay, so what they do is they have this expression matrix, then look at the first gene. So color-coded as this tail, the color light blue. And then we focus on the first genes and then try to use all the other genes to predict the first gene's expression. Okay, they just build this decision tree regression tools to predict values for the expression values in the first gene using all the other genes. And then they move to another gene, the second gene, use the first gene and all the rest of genes to predict the second value, the second gene's expression value. So they just repeat this procedure across all the genes. Okay? Look at the first gene and use all the other genes, build a tree to predict the second gene and see which other genes are the most important ones to predict the second gene. So after they do this, they build quite a lot of those decision trees. And from the decision trees, you can see, okay, which gene are most important factors to predict the first gene's expression. For the second gene, what other genes most important to predict the second gene? So they pick those important genes to make link between genes. At the end, they create this network. All right?
How can we get
The expression data depends on your setup. So here you have expression, then you have different conditions. But in single cell data, that you have all the expression and all the cells. Yeah. As I said, this method has been developed before the single cell age era. So that's why you have expression of the genes, and these are the experiment conditions. Just like experiment conditions, okay? Yeah.
Can we get data to create this prediction of the network in the website 10x?
10x.
Yeah, the website you showed.
Mm-hmm.
Can we get data to do this experiment?
Which website?
Yes, the
10x Genomics website, you can download example data, a matrix like this. Yeah, yeah. Yeah, so the algorithm design was for bulk RNA-seq. Yeah, but people found you can apply single cell data, apply it to single cell data. Yeah. As long as you have this expression matrix, you should be able to use it. So CINIC itself is not a gene regulatory network construction algorithm. It used gene 3 to do this network first, then using the database information to train the network. If you have a network, then gene 1 and 2 was linked by gene 3, but this relationship is not supported by the transcription factor and its target. This CINIC will remove this and make the network simpler. Okay, it only keep the database recorded relationship between transcription factor and the targets. The good thing about this is you remove a lot of connections which you just cannot explain by existing data. But the shortcoming for this operation is you have no way to discover new transcription factor and the target relationship. Then this is just try as a gene set whether those using a score to compute single cells with a network. But CINIC has this very complicated setup. So you have to install gene 3, you have to install package. I believe it's developed in Python. And also gene 3 is really slow. Okay, there are better implementation that's called GRE boost. Try to find a way, computational way to accelerate this gene 3 procedure. And in the channel, I gave a link. It's a YouTube tutorial. People from the CINIC developers, they gave a whole hours tutorial, see how you install and run CINIC in how to set it up. And so you can follow that if you really want to use this. There are several high impact factor impact journals have been published using this method. This is another one based on partial information decomposition. Okay. So information theory, in that case, we have to consider the entropy. They developed this package in Julia and published in 2017. And they claim that the computation is intensive. So this new high-performance computing language helped them to make the algorithm fast enough to apply to real data sets. So this algorithm inside, you have to consider the partial information decomposition approach. Okay, they have these equations. This algorithm called S-code. S-code is in R, and you have to give pseudo time. Okay, so this requires pseudo time to build differential equation somewhere in there. This is another. I don't know. I have never tried. Maybe they try to infer the causal gene regulatory network. They use the time series data and try to found, okay, this driver gene start to show its action, and then responder genes, which under the regulation of this key regulator should show its effect after certain time. So there's a time delay. So if you have a time series network, you probably have activation of this key regulator at an early stage. Then its downstream targets should have the effects have been shown off in the later stage of network. So they try to build this causal relationship between genes using this multi-layers information across time. So move to multi-layer network methods, and we can see what we can consider, what kind of problem we should consider. So one is the modularity change. You can have early stage, you have a module, and this module contains genes 1, 2, 3, and then this module changed, and the gene number 2 jump out, become a module member from another module. So if you have multi-layer network, you can do this module detection and see whether their time-dependent module change. This is one way you can consider to do the analysis. So this requires the time or layer information built into this Levin modulity detection algorithm. So you want to consider the change of the module membership based on which layer they are. So this equation looks more complicated than just single layer network problem. We want to look at those switch and some, or we call it network rewiring events. Okay, so it was no interaction between these two genes, but at a certain stage may have this interaction appeared, and maybe later it will disappear again. We hope we can learn through this multi-layer network structure to do this. Their method have been developed not for single cell, but if you Google time varying graph, they should pop up quite a lot of algorithm, maybe from engineering, maybe from other field of the network analysis. But I haven't seen too much about this time varying graph applications in single cell data yet. I believe the problem is the computational efficiency not enough to allow you to simulate or take thousands of gene cells into account.
So what's the biological meaning for the
Time where you can collect cells from a stem cell, then intermediate stage, then more derived development cell type. If you have a collection of the cells from different time points, you want to see, okay, early stage, this network looks like that. In the middle, and they're changed network, and in the later stage. So we want to cross the time point to learn those interactions or change of the interactions. This is a method based on non-negative matrix factorization applied to time series networks. Try to find those modules which conserved across time points. Some network modules, no matter what stage you are developing, they're still there. So this method try to find those sub-modules, okay? Now move to our own developed tools. You have already seen this a couple of times. So the idea for this single cell 24-net is try to compare two networks, and the two networks was constructed or derived from two single-cell RNA-seq experiments, and try to compare the condition, two different conditions. What we try to find is, in condition 1, we build a network, and then condition 2, we build a network, and look at the change. For example, G2 was connected strongly with G6, G4, G5, but in condition 2, this relationship changed. This connection become weaker, okay? We try to identify which gene has been differentially regulated the most in two different conditions. So this is the pipeline, and the one condition, two condition data, they contain the same number of genes, same set of genes, but from different cells. Then we construct the network using PC regression, and then remember I mentioned this denoise using tensor decomposition, and then reconstruct. Then manifold learning to do this manifold alignment to align two networks to find the genes, because the same genes appeared in network from condition 1, network from condition 2. So these genes, same gene appeared twice. And in their low dimension embedding, we will see regarding to the condition 1 and the 2, these two networks, whether this gene changed a lot in their position. Changed a lot means you just cannot align them very well, this particular gene in two conditions. So those genes will be prioritized and to see whether they response to the differential regulation. So we call the differential regulated genes. And as a group, see what are those genes responsible for, what kind of function groups are associated. Those genes will be used to tell the difference between these two conditions at the end. So this is an example we used to study the morphine injection response in the neuron of the mouse. So those are key genes, and they're supposed to change the expression, but the DE analysis, you just don't see any difference at all. This is like 3 or 4 biological replicates. 4 biological replicates, they're all neurons. And this is genes, this particular gene's expression before injection of morphine and after. No difference at all. And then you have to go through this pipeline, and they found this particular module and they changed it a lot. Not by the average expression, but it's by the interaction between genes. And it was like that, but after the injection, the response looks like that. The change, we can detect this module. And when you put all these genes together, and you can see those genes are enriched for the function of morphin, addiction, opium, signaling, GABA A, B, receptor activity, G, protein signaling receptor. So all this response tells us this is true because we know the challenge, we know the response is supposed to have. So that's validate our method, which detect the subtle difference in their regulation change rather than the average change of the expression. Then follow that, we see, okay, we can compare knockout and the wild type to see whether the function of the knockout will change this gene function. So the same pipeline here, exactly copied from the single cell 24-net. In this case, we are not doing two conditions. This is called a virtual knockout tools. The difference is we do not require two samples. We don't need this sample. We only need one sample. This is from a wild type, okay, normal condition. You sample the cells and using single cell RNA-seq to get thousands of cells expression level in thousands of genes. That's the only matrix we need to do this analysis. So what the pipeline will copy this, okay? Copy this wild type expression matrix and put them side by side. If you run this pipeline right now, you will still get some genes, okay? So you know those genes are false positive because your input two are the same. You should get some random genes because some random number generator was inside in the pipeline. You still get the gene list, but those top ones will just no function, no meaning. But what we did, we just found one genes and set its expression to all zeros, okay? Now these two matrices are differed only by one gene's expression. It was expressed the gene, but now we artificially set the expression of this gene to all zeros in all the cells. So this simulates a condition which this gene has been knocked out without expression at all in that condition. So we just compare these two matrices and just run this pipeline again. It will give you a list of genes. So always on the top, the first gene will be the gene you knock out. And then we are interested in the second gene and the third gene, all the genes next to the first. Okay? So next to the first, all those genes are, after you knock out this gene, what other genes will follow the expression of this gene change, will also change their expression or response or their position in the network after you do this. So this is a virtual knockout tool. So we try to knock out a gene and understand its impact on which other genes, use other genes' information to predict the function of the gene you just knocked it out. So this is a simulation. We knock out TRIM2 genes from wild type mouse. Okay, and to find the top genes which supposed to be impact after you knock out these genes, and then look at the function enrichment of those responder genes. What we found is this will be genes related to the Alzheimer's disease with lysosome, cholesterol metabolism. And later we know, okay, the TRIM2 genes are really associated with some of those functions we know about the TRIM2. And also, they're real knockout mice, so we can get a real knockout data and to compare with wild type to study this function in real animal knockout. And we found the same pathways in real knockout animals. So this tells us the simulation probably should go first before you go to the animal studies to predict, give you better hypothesis what you expect to see, rather than do the animal study first, then discover almost the same thing. And this method, we can delete any genes. We can delete 2 genes at the same time or 3 genes at the same time. Yeah. So the whole idea is based on the network. You build a good, more realistic network, and then any perturbation on the network you know at which position you actually perturb the network. You found the neighbors. And also, this virtual knockout can be done for all the genes, okay, one by one. So you build a response landscape, okay. For instance, you knock out gene 1 and 2, their responder genes are similar, so you can infer that the gene 1 and 2 have a similar function themselves by looking at their perturbation profile. At the end, this is an interaction study. It's also based on the network, okay? So here is a network built from cell type 1, network built from cell type 2, and their corresponding networks, so we can, to do this manifold alignment, this is just using network to do this. So this network, after alignment, we found the distance of a pair of genes, gene I and J distance. Will be, one is a ligand and another is corresponding receptor. If they're close to each other, indicate they probably interact between these 2 cell types. So again, in the middle of this, we still rely on cell type specific gene regulatory networks to offer this background in order to allow the gene interact based on this background of regulation to cross cell types. That's all. So I just summarize. We start by looking at the centrality measurements, and then WGCNA, and then jump into single cell data and advantage to do the gene regulatory network. Gene 3, and also I let you consider multi-layer network analysis. This not well studied. I haven't seen many good tools or algorithms have been developed, but should be a good field for people to dedicate efforts on. And then I introduce these multiple tools in this suite. Hopefully you get the idea and apply all this in your own studies. Okay, any questions? Okay, great. It's been 13 weeks, and from early this year to nowadays, I hope you have a good understanding of all this global framework from a matrix multiplication until today and network. All right? Yeah. Let's see what you will have next week. Talk about these projects, okay? Yes, stuck again?
Yeah.
Well, yes.
It's getting
It
stuck.
shouldn't have.
So I had a question. So if it gets stuck,
Mhm.
Like I filtered out everything and I was doing DE analysis.
Mhm.
For processing, nothing green came up. Like it wasn't happening.
You shouldn't. For example, I show you the example data if you use. Yeah. So, you know, if you can run the example data, then you should be able to run other. If you do the DE.
Oh, I tried to filter and everything was done, and then I was doing DE,
Yeah.
and
So
it
if
did
you
not
see,
process anything. It just broke
you
down.
see it.
Maybe the data is too big.
Yeah, I don't know. Maybe. Yeah. You see this? It should work. There's no reason.